So today we are talking about fMRI some more and getting more into different kinds of experimental designs and how we analyze the results of those different kinds of experimental designs. And hello, clicker. There we go. So I showed you this slide before. This is the data from one of the first fMRI experiments that was ever published in 1992 by Ken Kwon and colleagues. And they used what we now call a block design, which is where for a certain period of time subjects were seeing a particular kind of stimulus, and that was a fairly extended period, in this case 70 seconds, as you can see here. So it was off for 70 seconds first, that's kind of like their baseline or control period, and then on for the next 70 seconds, and then off again. And this was simple visual stimulation. So just like a black and white checkerboard on the screen, and then off and on again. And they did that because the early studies that have been done prior to really doing it in humans, and even early human studies, suggested that the changes that they were measuring were pretty slow. They weren't fast changing like EEG. And so this was a way of sort of being able to stimulate over time periods that seemed to match what they were observing empirically in the data. And like I say, this came to be called a boxcar design or a block design. Boxcar because it kind of looks like a bunch of boxcars as part of a train. So the simplest kind is just alternating between an on and an off condition, or a target condition and a control condition. So off in the original case meant, physically the stimulus is off, and then on meant you saw a stimulus on the screen. But you can imagine lots of block designs where you've got tighter control. So for example, if you want to look at brain areas involved in processing visual motion, you could show moving dots as your on condition and the same dots static or not moving as the off condition. So it doesn't have to be no stimulus. It can be, and ideally it should be better controlled than that. And the motivation or the sort of thinking behind that actually goes back way before brain imaging was ever invented, to F.C. Donders, who is a Dutch psychologist and researcher. He has this quote, "The idea occurred to me to interpose into the process of physiological time some new components of mental action. If I investigated how much time this would lengthen the physiological time, this would, I judged, reveal the time required for the interposed term." That's a lot of very flowery 19th century language, but basically when he's talking about physiological time, he's talking about reaction time. And so he was one of the early psychologists who was using reaction time as a measure of cognitive processing on the very obvious and simple assumption that the amount of time it takes you to press a button in response to some stimulus reflects the amount of time it took your brain to process that and then send the signal down to your hand and press the button. And then the question became, how do we look at the timing required for different sub-processes, right? So in a simple case, like I say a word and you have to press a button or say it back out loud, there's a lot of processes in the middle there, right? So I said it, you have to hear it, it has to go from your ear to your brain has to make a sort of response, detect it, and then send a signal to your arm and send that down and press the button. So there's a whole different series of events going on there, and we can't necessarily break that down more simply, because if we have the button press, we need the motor response, we need some sort of stimulus processing to trigger that motor response. But the idea is if you interpose additional processes in the middle, the amount of extra time it takes you to make a reaction is the amount of time required for that process. So if instead of just repeating my word, you had to do something like I'm saying a noun, say I'm saying a noun and you have to come up with a verb. Or I'm saying the name of something and you have to decide if it's alive or not, what we call now an animacy judgment. Then you could basically say the additional time required to make that judgment, as opposed to just repeating the word, reflects the amount of time that your brain took to do that particular process. So this is known as Donders' subtraction method, and just formulaically, if you imagine task A is components X to Z, so perception to action, and then task B is X to Z, but with Y interposed. So Y would be that animacy judgment or coming up with a verb or whatever. Then the difference in the reaction times between task A and task B, between when you hear that word and when you make the response, is the time required to perform action Y. Actually, I'm not quite sure of my order of slides there. So Donders showed this for behavioral research, And then when people started doing brain imaging research, even before fMRI with PET studies, the same thing became relevant, because just like if I just have you do a task and measure how long it takes you to do the task, it doesn't tell me how, which parts of that task took how much time. If I throw you in a PET scanner and inject some radioactive tracer into you and have you do a task, you're going to get that radioactive tracer distributed throughout your brain, but you don't really know what about that is different from if you weren't doing that task, or if you were doing some other task, right? You need some basis for comparison. If we don't have a baseline, we don't have anything to compare to. And ideally, those baselines are as tightly controlled as possible. So again, if I'm just lying in the scanner and I'm staring at a black screen, and then I see a black and white checkerboard, then it's fairly simple, the task, right? It's just perception. I'm not required to make a response or anything. So that's reasonably well controlled. So it's sort of visual stimulation on, visual stimulation off. But say I wanted to look at areas specifically involved in face perception as different from other kinds of visual perception, then we have to worry about the fact that faces are a visual stimulus, but other kinds of visual stimuli would also activate occipital cortex probably similarly to faces. But there might be areas that are specifically sensitized to faces and not to household objects or animals or trees or other sorts of things. And that was actually one of the earliest areas of investigation with fMRI, with visual perception. So Nancy Kanwisher and colleagues at MIT and Harvard did these studies where they presented people with faces, and they actually compared them to multiple different control conditions or baselines to see what was sort of consistently activated by faces but not by other kinds of visual objects, and using this kind of block design. So here they're comparing faces to various kinds of objects. So you've got, I don't know actually what that one on the left is. Kind of looks like corn to me, and a telephone and a crab. And then there's the areas that light up more for faces than for other objects. And the sort of yellowy spot there is in the fusiform gyrus in the right hemisphere. This is one of those classic radiological convention maps I might have mentioned before, where it's like you're staring at the person from their feet. So the left side of their brain is on the right side of the image, and vice versa. So even though that's on the left side of the image, it's the right side of their brain. And we already knew that the fusiform face area was probably involved in face perception, which is why we called it the fusiform face area, from neuropsychological studies of people with prosopagnosia, like the Oliver Sacks book, The Man Who Mistook His Wife for a Hat. So it was known that this is probably where we're going to see something lighten up, and sure enough it was, and more on the right than the left. That was consistent. So that was nice. And you can see here the block design. So the gray bars are showing where the faces were presented, and then the whites were when other objects were presented, and the dots are just black screens that were interposed in between those different blocks. And you can see that it's not like this area doesn't light up at all for other kinds of visual objects, but relatively there's more activation in the face condition than in those other object kind of blocks. So that's nice evidence that this area is related to face processing, probably. We can do the study again, exactly the same study, so just straight up replication is always good scientific practice to show that something works, and we get a similar pattern. Then here they changed the stimuli a bit, so they made the faces black and white instead of grayscale, and then as a control stimulus took the black and white components and just sort of scrambled them in different positions in the image. Again, show greater activation for the intact and the scrambled faces. Here they compare faces to other body parts, namely hands. Again, more activation for faces, faces relative to houses, and then changing the task to now be, I forget what the task was for the other ones, if there even was a task, but here they did a one-back where you have to say, is the stimulus you've seen now the same as the one you saw immediately before it? So again, the idea there is to show that this area is responsive to faces regardless of whether you're paying attention to the faces or doing a working memory task, or whether it's task independent or not, and sure enough it was. So that's one way of doing block designs, is just alternating your target condition with some other control condition, and then you can you could do this within the scanner, within an individual. So do like each of these could be 1 5-minute run in the scanner, or you could do multiple experiments across different individuals. It doesn't really matter. You can do it either way. But it's using this method of subtraction where you're trying to control for different properties, because it's sometimes hard to control for everything, right? So with faces, the scrambled faces might be the best control in the sense that it's all the same sort of visual bits. They're just rearranged on the screen, but the overall intensity, luminance, and all that is the same. You could argue that faces versus hands is good because that's isolating faces relative to other body parts. It's a more sort of higher order thing. So you can see there's not necessarily any one best control condition. So by doing this sort of looking across multiple control conditions, that's one way of addressing this with using the subtraction method. Now with the subtraction method, you don't have to restrict yourself to just a single sort of contrast within a run. You can actually have different types of on blocks and different types of off blocks. As an example of this is a study that I actually did as a postdoc at the University of Rochester. That image is Ted Sapala, one of my collaborators, who's a deaf native sign language user, American Sign Language. And in this study, we were interested in comparing sign language sentences that conveyed the same grammatical information in different ways. And American Sign Language actually has three different, excuse me, I should rephrase that. It has different ways of expressing things. So for one thing, the simplest way to express grammatical relations, like who did what to whom, is just using word order, which is what we use in English, right? So John kissed the dog is different from the dog kissed John. And that's true in sign language as well. But another way is, like some other languages, like German, there's case marking. So in sign language, at least in American and many sign languages, you can use the direction that a sign moves through space as an indicator of who's doing what to whom. So the verbs, you make a sign for a verb, so like this is a sign for help, and moving from me out to you means I help you, moving from you into me means you help me. And so the directionality can convey things independent of the word order. So we were interested in contrasting those and also sentences that used more rich sort of storytelling devices in sign language. And because each one was different, and I'll show you the video in a sec, each one was different. So like the word order one, you're just signing things and everything's sort of occurring in the same space in front of you. The one with that spatial syntax, as we might call it, involves more movement through space, right? And so directly comparing those two, you've got the same sort of content verbally, linguistically, with that difference in how the grammar is being expressed, but one also just visually has more visual motion than the other. So rather than directly comparing them, we wanted to have a comparison, a control condition for each of those conditions that was matched to it by playing those sentences backwards and actually overlaying three at once. Because it turns out when you play sign language backwards, it's kind of like if you watch a movie backwards, stuff still makes sense. It's human action. So we basically had three different on conditions and three different control conditions matched to each of those. So just because it's sort of fun to visualize these. So that's the word order one. And he's describing the sentence as something like, some people watched a car accident and they ran to help. And there's a bit more motion. It's not overly clear, but in this one there's a lot more. You can see like the facial expression and that sort of thing. So that's more how a signer would naturally express themselves. But because it's so rich in terms of like the facial expression and the movement, that's not the way people typically use linguistic stimuli. So we wanted to have both. And then here you see the control stimuli. So it's not the best control stimulus in that it's actually got like more stuff going on and more visual motion than our on condition. But the rationale there was you can still see the face, you can still see the hands moving, all of that stuff that we want to control for is there. And any areas that are actually sensitive to just like biological motion or face perception are going to be more excited by those control stimuli than our on stimuli, our target stimuli. So it sort of works in our favor. So other areas might show increased activation in the control condition. We don't care about that. We don't have to look at that. But it should be like more than compensating for anything in the on conditions. So really the point there is just that your block designs can be quite complex, and you get different kinds of on conditions, different control conditions for different of those on conditions, and so on. Sort of a step above the subtraction design is what we call an additive factors design. And this is actually now sort of going back in time. So this is the original PET. Oh, sorry. Question?
Yeah, question for sign language. Is
Yeah.
it presented in the brain for people that speak sign language
Mm-hmm.
as our spoken language? Like it's in the same brain area?
Yes, sorry. I totally like ignored all the actually cool cognitive neuroscience stuff about that study. But yeah, so even before we were doing fMRI studies, neuropsychological studies of people who knew sign language and had strokes, suggested that it was represented in similar brain areas. So left-sided stroke leads to aphasia in sign language, just like it does in spoken language. When we started doing fMRI studies, the same thing was borne out. And so sign language tends to engage those classical left hemisphere language areas like Broca's area and Wernicke's area, just like spoken languages. In some cases, it actually engages the right hemisphere more. And that's why in this one we included, so historically, we did some studies originally with much more naturalistic sign language, so like that last condition you saw with more facial expression and body movement and so on. And we actually saw a lot of right hemisphere activation for those sign language users compared to spoken English users. But up until that point, at least, everybody who had done spoken language had actually used written English or written German or whatever as their stimulus. So you're comparing like printed white font on a black screen to this very rich stimulus with all this sort of prosody and affect and that kind of thing. And so in this study, we had the more sort of stripped down sign language compared to the more prosodic facial expression stuff. And sure enough, a lot of what we saw in the right hemisphere was due to that. And so when you strip that away, it looks much more left lateralized, like typical spoken language. Yeah. All right. Anyway, so additive factors is just sort of a step up. So rather than the sort of subtraction between two things, you have multiple levels. So another term that's used in other areas of psychology sometimes is levels of processing. So in the original PET imaging studies that were done in the late 1980s, it was actually Mike Posner, who was a cognitive psychologist, who brought this design to the other people working in that group, who were radiologists and MDs and knew sort of the brain imaging side good, but not the cognitive science side as well. And so the idea was to look at brain areas involved in reading and sort of simple linguistic processing, and recognizing that when you read a word like board there, the bottom right panel, there's a lot going on, kind of like I described before. If you start breaking that down, you're seeing a visual stimulus. It's letters, so it's a specific kind of visual stimulus that it's composed of like lines, basically, in particular patterns. We as literate human beings, we know what those letters mean. They have sounds to us, so at least something like C over there, tweel, you can pronounce letter strings even if they aren't words. So you've got these different levels of sort of what linguistically we call orthography, which is the printed letter forms, phonology, which is the sounds. So compared to like B, which is just consonants, you can't pronounce that easily unless you imagine vowels stuck in there, whereas tweel you can, so you've got the phonology. And then we've got semantics, or the meaning of the word, which is when you go from C to D. So the idea was, rather than compare board directly to A, which is a sort of false font, so it's a visual control for printed words, but if you just compared A to D, you'd have the visual, the orthographic, the phonological, and the semantic. So the idea was, rather we build it up and we compare consonant strings to false fonts, and then pronounceable words to consonant strings, and then meaningful pronounceable words to meaningless pronounceable words, and see what differs between the brain activation in each of those things, and isolate those cognitive processes separately. So that's the additive factors approach, is basically sort of, rather than just comparing two conditions, you compare kind of a hierarchy of conditions where you're adding on different things. So that ASL one that I showed you before was sort of an additive factors design, in that we have the straight up word order way of conveying the grammatical information. Then the ones that use the spatial devices are still using word order. Like, word order is still present in the sentence because there's multiple words, so they have to be in some order. So you've still got that plus that additional grammatical device. And then the third type had that spatial grammatical device plus the other stuff. So another example of kind of an additive factors design. Make sense? Okay. So the one limitation of these additive factors designs is they assume that what you, as a human, what your brain is doing between those conditions differs only in the ways that you think it does, basically. So formally, this is something that Donders called pure insertion, which again is sort of flowery language, but it basically says that when we insert Y, any changes we see in reaction times or brain activation are due directly to Y, and they don't affect X or Z, which are the sort of pre-existing processes. So again, in the sort of simple example of we hear a word and we have to make a verbal response, we insert the task of making a judgment around animacy and saying that out loud in, and we assume that doesn't affect our perception or our response ability. In some cases that makes sense, but in other cases you might get interactions. And one of the classic ones that people worry about in brain imaging is attention, or just sort of more simply level of interest. So here's a face stimulus on the left, and on the right is a different kind of scrambled face stimulus, which has just had a transformation applied, where it's equal in luminance and the amount of black areas and gray areas and white areas and so on, but it doesn't look like a face. So as a visual control stimulus, it's very tightly controlled, but as an attentional control stimulus, not so much, because faces are way more interesting to look at than these other things. And so if you're just sort of lying in a scanner and you're seeing a bunch of blurry images, you're likely to get bored and pay less attention to those images than if you've seen a bunch of faces of, at least mildly attractive or interesting. They're just faces, they're interesting, right? So the level of attention you're devoting could actually contribute to the differences in brain activation you see between those things. So that would be considered a violation of pure insertion. There's no magic sort of formula for solving this issue. It's more, as with any experimental design, you just have to think through in detail all of the things that might be different, and not just the stimulus features and sort of physical properties, but actually the more cognitive attentional things. Another one that people worry about a lot is difficulty. So if you have people, this is not the best example, but if you have people doing some task, like a memory task, some people might have a bigger working memory span than others. And so if you're giving everybody the same number of items to remember, some people might show stronger activation in working memory related brain areas because their span is smaller and it's just a harder task for them. And that becomes magnified if, say, you're comparing children to adults, or like older healthy adults to older adults with mild cognitive impairment or Alzheimer's disease or something like that, where now you have this chicken and egg problem that you don't know. You know the task is harder, and that's why you're interested in scanning their brains when they're doing a memory task, but is any difference you see because they're working harder, so it's more sort of a cognitive effect of their condition, or is it the cause of why their condition is there and why their memory performance is worse? And those things can be difficult to deal with. One thing that people have tried is you can give multiple levels of difficulty within a task to each individual, and then you can compare, the same actual condition or same level of same sort of overt level of difficulty. So like a working memory task, you'd say compare the retain 3 items in working memory between the two groups, look at that. But then also just compare based on actual performance levels, right? And so if one group is achieving like 80% accuracy at 4 items, and the patients are only, can only achieve that accuracy with 2 items, then you compare those two conditions that differ in the number of stimuli they're holding in memory so that you're equating for difficulty. And then you can look at, when you equate for difficulty, are there still brain activation differences? So there are ways around it that way. All right, so that's the block designs. That kind of got us through the 1990s, but people quickly became frustrated with the limitations of the block design. And this was true even from the early days of PET. People recognized, because PET imaging, you have to do like 90 seconds of one task, stop the scan, wait for the radioactive tracer to flush out, and then do another one with a different condition. So it's like even worse than fMRI. But the problem is that block designs don't allow us to do things like randomize the order of trials between different conditions. We can't do like an oddball, like remember with ERPs, P300 kind of experiments where you have beep, 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 boop, and you see an effect of that deviant stimulus. If you're doing a block design, it's not really an oddball because the first stimulus of the second block could be an oddball because it's different from the previous block, but it's not an oddball after you present 2 or 3 of them. So that's an issue. If you want to look at brain activation relative to the kind of response somebody's making, right? So you imagine like from trial to trial, some trials are error trials, some trials are correct trials. If you want to separate out brain activation when people are actually getting it right versus when they're getting it wrong, you can't do that if everything's smushed into a big block. Or multi-phase trials. So in a typical working memory experiment, you'll have 3 phases. So there's the encoding phase where you present the items to the person. So say it's a list of letters. Then the retention phase where they have to retain that information in their working memory. So that could be like 2 or 3 seconds of encoding, maybe 10 seconds of delay, and then a response period where you either prompt them with one of the letters and say, was that in there, or have them repeat it back or whatever. So there's 3 different phases, and if you want to look at brain activations separately in each phase, that's hard to do within a block design because your block is going to be like a bunch of those trials back to back. It's hard to separate. So people were really looking for ways to get more sort of fine-grained analysis of the fMRI time course, realizing that you're at best, certainly in those days, even now, the TR, so the time it takes to acquire one whole brain image with fMRI, was about 2 to 3 seconds. Now you could do it probably in 1 second. 1 to 2 is maybe more standard, but still it's pretty slow compared to ERP. But at the same time, if a block design is something like 30 seconds long, then you've got somewhere between 10 and 30 samples in there. So what if we could actually break that down and look at more fine-grained differences in there? The issue was that people felt that the signal was pretty weak, and so those blocks were required to sort of have things rise and come up to a decent level to be able to detect. And also the fact that, as I talked about last time, the hemodynamic response function that we're measuring is actually pretty sluggish, right? So it takes 2 to 3 seconds to rise, 6 to 8 seconds to peak, 10 to 15 to maybe 20 seconds to come back to baseline. And so if you want to be able to isolate the HRF to an individual stimulus, you basically have to space your stimuli out at something like 15 to 20 second intervals. And that's not very efficient. Remember, fMRI costs $500 an hour. So all of a sudden you're presenting very few stimuli per unit time. That gets very expensive. It's incredibly boring as somebody being in the scanner to see like a face and then wait 15 seconds staring at a black screen and then another face. So it's not an ideal way to do it. Some of the early studies were done like that, but people quickly figured out that there were tricks that they could use to present things more quickly and still be able to separate out those hemodynamic response functions to individual stimuli. I couldn't figure out where the best place to put this was, but this is one of the original, one of the earliest event-related fMRI studies where they're looking at responses to single trials. And here it was a memory task where people saw a bunch of, I think, words in the encoding phase. So not working memory like described before, but just a whole 5 minutes, you're just seeing a list of words. You're supposed to remember them all. And then you do a test after you get out of the scanner as to which words you saw. And they'll sort of show you the words you saw plus some FOIL words, and you have to say which ones you saw. So when they analyze the data, they analyze separately in that, when you're in the scanner doing that encoding, those individual items that they saw and later got correct, so they later remembered them, compared to the ones that they saw but they later got incorrect, so they didn't remember seeing them. And they showed that across a number of brain areas, what you're seeing here, the purple over the green. So the purple is remembered trials, the green is the forgotten. So there was consistently stronger activation throughout a number of different brain areas for the later remembered items than the later forgotten ones. And that was only possible with event-related fMRI, because again, if you just analyze the block, you're going to make some correct and some errors within that block. Okay. So here's the challenge. So the top panel here is so those sticks, the thin vertical lines, are showing you when the stimuli occurred. And here the SOA, so the stimulus onset asynchrony time between the onset of one stimulus and the next is 9 seconds. And then that's our model hemodynamic response function. And so you can see stimulus, HRF rises, falls, basically comes back to baseline before the next one. So that's what we call a slow and inefficient event-related design. As you start to pack them close together, they start to summate. And basically these things, and people have measured this empirically, they summate linearly, meaning that it's just literally the sum of, where this first one was plus where the second one is. So you can predict it just by sort of overlapping them and adding them together. So with 6 second intervals, you can see you still get this nice variation. So you could, in principle, pull out the responses to each individual stimulus. But as you get to about 3 seconds, the problem is that you don't have much time for the HRF to drop down before it comes back up. And this is important because the way we analyze the signal is to look for systematic variance in the amplitude of the signal as a function of stimulation. So here we can see a brain area that's sensitive to whatever we're doing as a stimulation should show this up and down pattern of activity, whereas an area that's not sensitive should more or less show a flat line. With the SOA of 3 down here at the bottom, the problem is that aside from this ramp up over the first few seconds of the experiment, the activation or the bold response in this active area is basically a flat line. And so it's hard to know how that flat line is different from the flat line in a brain area that's not responding at all, because the only difference between them would be one would show this early response. And so that's, you have sort of one part of your scan to capture, and the rest is not very useful. So you really need some way of introducing more variability in the predicted brain response. This just sort of emphasizes the same point, but with more different examples of timing. And you can see as you get out to like 1.5 second spacing that it literally is just a flat line, and there's no way you could get that done. And the saving, okay, actually, before I get to the saving grace, this is basically what a block design is in little chunks, right? So a block design is you pack a bunch of stimuli together, and the whole goal there is to pack them close enough together that the bold response rises and stays high over the duration of that block and then comes back down. And there it's actually better to have your stimuli packed tightly together, because here you can see these are all in the same amplitude scale. When you space them farther apart, you have less stimuli in your block. You're stimulating the brain less, and you get less overlap. So as you pack your stimuli in tighter with a block design, you actually get more returns. You get a stronger signal. So that's great, as long as you have those off periods in between. But the problem is you can't isolate still the effects for individual stimuli within that. So the saving grace is what we call a jittered event-related design. And here we just randomize the spacing between the stimuli. And so you have some that are packed closer together, but then you'll have gaps where they're spaced farther apart. And here you can see in the top two panels, let's look at the second one first. Here's 120 events with this fixed spacing of 1 second between the events. And again, HRF rises, and then it's flat throughout the whole thing. However, if we have exactly the same number of events, same duration scan, just by randomizing the spacing between them, we're going to introduce some longer gaps between some stimuli, as well as some sort of mini blocks where they're packed closer together. And now you have very systematic variance in the bold signal. Now, it doesn't look like a block design, but if we know what the shape of the HRF is, which we do, and we know when each stimulus comes on, which we do, we just sort of design a prediction in terms of how the brain activation is going to look based on putting one of those HRFs at the start of each stimulus, figuring out how they overlap, and you get something like that. And then you just say, okay, which parts of my brain show a correlation in their bold signal with that weird, whatever, semi-random looking kind of pattern. And by chance, although that's sort of a weird looking pattern, brain areas that aren't systematically related to your stimulus shouldn't show exactly that kind of variance. They might show weird other random patterns, but they're not going to show that one by chance or with very low probability. The cool thing about these jitter designs is that just like block designs, they actually work better when you pack more stimuli in, even though in the evenly spaced design that's worse for you. So here we've got 80 events, so 40 events less, basically like 2/3 the number of events in the top. And by jittering them, we still get this nice random variation that we could analyze. But overall, if you look at the amplitude of the activation, the max there is about 1.5, whereas in the one with more events, the max is 2 or even 2.5 at different points. So by packing more in, you get some of the advantages that you get with the block design and packing stimuli in, but you still have this random variance that allows you to, or systematic variance, that allows you to analyze the signal and differentiate the brain areas that are related to your condition with those that are not. Okay, so that's the event-related designs, and those are quite common. Now, it's not the case that you necessarily have to use an event-related design, because as I showed you, the block designs are very good at packing a bunch of stimuli in and giving you a big, strong, bold response. So if your experiment lends itself to blocking the stimuli and conditions, that's absolutely fine, and you probably actually get stronger results, more statistically robust results using that approach. But if you can't, then you need to use an event-related design, and there the only thing is to understand that you should use the jitter and not just evenly space out your trials. So the next issue that we're going to talk about is that of baseline conditions. And this starts to, it almost sounds a bit hair-splitting, but there's a difference, certainly in the way I use these terms, between baseline and control conditions. So a control condition is a condition that's designed to control for stimuli in your on condition that you don't care about, or stimulus factors or task factors, right? So if we have faces, our control stimulus is something like a scrambled face. Okay, a baseline condition, the baseline is really sort of what that brain area is doing when it's not active. Now, you can get kind of philosophical about what that means, right? So if I put you in an fMRI scanner and tell you do nothing, you're not literally doing nothing unless you're like a Tibetan monk and you've been meditating for many years and you can sort of shut everything down. Your brain's still going to be going, but you're not going to be getting outside visual stimulation. You're not going to be making decisions, making motor responses, or anything like that. So that is basically considered sort of the best baseline condition in terms of figuring out where brain activity would be within a certain brain area without anything else going on. And it's important to have these in fMRI designs for the most part, with some exceptions, because of what's described here. So imagine that we just have our block design, we have the on condition, the off condition, so faces, scrambled faces. We find a bunch of different areas in the brain that show a stronger bold response for the faces than the scrambled faces. Our default assumption would be something like the first pair of bars here, where blue is faces, green is scrambled faces. So that basically tells us, you know, this area is involved in face processing, shows activation for faces and sort of weaker activation for scrambled faces. But it could be this. So here, the difference between the height of the two bars is identical to here. So it's the same magnitude difference. So if we just do the subtraction, this and this are going to look the same in our sort of statistical map. But here, obviously, there's a difference because if 0 is our baseline of staring at a black screen, then what this is telling us is that faces activate that brain area kind of weakly, but the scrambled faces actually suppress the bold signal in that brain area relative to seeing nothing. And although that might sound counterintuitive, like if the brain's already sort of at rest, how can it go down from there? But at rest, in this case, again, your brain, like you're not doing nothing. Your brain areas are doing something. And maybe some brain areas are actually doing something when you're not doing anything else that they're not doing when you're engaged in a task. So there's sort of boredom-related areas, whether it's like, you know, internal speech or planning or just sort of paying attention. Maybe like you're paying more attention to the sound of the scanner because there's nothing distracting you from that. So other stuff could be going on. So you could see that decrease. And critically, if this dashed line is our threshold for significance relative to baseline, this difference looks like it's a significant difference between the experimental control conditions. So the magnitude of the difference here is sort of biggish, but the experimental condition itself relative to baseline isn't actually significant. So that would be a really bogus kind of finding in a paper. And you could end up with like your face-related areas plus other areas that have nothing to do with face processing. And the other ones really just sort of take that same idea to a greater extreme. In all cases, I made these such that the difference between the green and the blue is exactly the same, right? So it's, all 4 brain areas are going to look the same in that direct contrast map. So you need some way of including that baseline condition in there as a way of isolating things. And this, I didn't really highlight it, but in that original face processing study that I showed you, remember I said there were face conditions, objects, or whatever the control stimuli was, and in between each one there was that little sort of baseline period. So they included that, and that's fairly common. In other cases, people don't do that for a couple of reasons. One is that if it's something that's very well established, like face processing, sometimes people do like a face versus scrambled face control block design as what they would call a localizer run at the start of a study. So say you wanted to investigate some response properties of the fusiform face area. So, you know, we have this area. So for example, some people early on were interested in this question of whether it was face specific or it was related more to visual expertise. and we're all kind of face experts because we look at faces a lot. We have to differentiate between individuals. But in fact, it turns out that the fusiform face area shows stronger activation for bird faces relative to control stimuli in people who are birders and recognize and differentiate between these birds, and same with car experts. So in that case, you want to first isolate the FFA, the face area, by doing that block design. And because people have already said, okay, we know the fusiform face area responds more strongly to faces than scrambled faces, we know it's in the fusiform gyrus, all we got to do for any individual is show this contrast and zoom in on that area. Don't worry about anything else. So that's an acceptable case where you could have that sort of on-off contrast and still be okay. But if it's exploratory, especially, and in a lot of other cases, you want that additional baseline condition. And the way you would use it in analysis is doing something called masking. And so in this case, you're basically doing a pair of contrasts and looking at what's kind of consistent between them. This is another study that I ran. I use lots of examples of studies I ran because it's very easy for me to like pull up the data and make the figures. So it's not so much an obsession with my own work as just it's convenient. So here we were looking at gestures relative to control movements. So the gestures were like meaningful gestures like thumbs up or A-OK or something like that, compared to other hand movements that didn't have any conventionalized kind of meaning. So the red is our gestures versus those control movements, anywhere that showed that difference is statistically significant. And then the blue is gestures compared to the baseline of just staring at the black screen. You can see, as you might expect, the blue is pretty extensive. Includes all of like the occipital cortex, parietal lobe, lots and lots of areas, because it's not a well-controlled stimulus. It's like showing a person with hands and all this stuff relative to just a black screen. So lots going to light up. But the critical thing is to look at what's consistent across these gestures relative to the baseline and gestures relative to the control stimuli, because then we know that they're active relative to that baseline, so it's not any deal with the suppression, and bigger than our control stimulus. And sure enough, you can see, so those are the purple areas. You can see that a lot of these areas in red that showed the gesture versus control significance actually weren't activated for gestures relative to baseline. So they're not, whatever they're doing, it's not what we would want to isolate and say that's a gesture processing area. It's only those purple ones, which are sort of a small subset of the overall activation pattern, that actually relate to what we are interested in studying. So that masking process is an essential step for most fMRI analyses to really isolate brain activation you care about. Quite closely related to that is this notion of conjunctions. So in a sense, what I showed you before was a conjunction between the gesture versus baseline contrast and the gesture versus control contrast. More generally, a conjunction is looking at what's active in the brain across two different contrasts. So sort of symbolically here, this first one, A greater than B plus C greater than B, is kind of what I was showing you before. If A is gesture, actually that's not right, never mind. It's slightly different. But here it's like we have one control condition and two different active conditions, A and C. Or you could have different things, so A versus B, so A versus its control condition, so like the word order sign language versus control for word order, and then the spatial syntax sign language versus control for spatial syntax. And then when we look at the conjunction of those two, that tells us sort of what's consistently activated by sign language, irrespective of how that grammatical information is being conveyed relative to control stimuli. So it's an additional step that helps you sort of make interpretations about brain areas, because you can think more broadly that if we're interested in something like, say, semantic processing, we could define semantic processing in a lot of different ways, and we could come up with a bunch of different contrasts that did that. And those might each have their own weaknesses in terms of our ability to precisely control for all aspects of the stimuli that we don't care about. But if we have multiple different ways of sort of defining and exploring some semantic processing, then you can use the conjunction analysis to see what's consistent across all of those different tasks that we call semantic, and what sort of falls by the wayside because it was present in verbal semantic processing but not visual semantic processing or something like that. In this example here, we did an experiment where we had people they were seeing words on a screen. They either had to read them aloud, say the word yes instead of reading the actual word aloud, which is a motor control, so they're doing motor task and hearing themselves speak but not reading the word aloud, or silent reading. And these things, the produced condition leads to better memory later, which is what we're really interested in. So here we've got the contrast of produced versus that sensory motor control, which isolates certain brain areas, motor and auditory areas. Produced versus silent reading, which kind of has overlap but much more extensive, which makes sense because here we're not controlling for either hearing your own voice or the motor aspect of producing speech. And so the conjunction analysis here shows us what's common to the production condition relative to both of those control conditions. So it helps sort of isolate what's unique to that one condition by controlling in different ways. So conceptually, you can think of conjunctions in terms of a Venn diagram. So the conjunction is the overlap between the areas that are active in one condition or contrast and the areas in another. And then the disjunction, which is a little less commonly used but can be useful, is the areas that are unique to one or the other, but not in the overlap. So there, if you wanted to isolate what's sort of specific to one condition and not in other conditions that might share some overlap in processing, that's where the disjunction analysis would be handy. Okay, and I'm going to cover 2 more slides and then we'll take a break. So the fMRI adaptation design came about because people were interested in looking at activation within brain areas and thinking about the fact that within an area that sort of lights up as a blob of activation for, say, faces, there might be subpopulations of neurons within that area that respond to different properties of faces. So some might respond to face identity, some might respond to the orientation of the face. If you think more simply, like low-level primary visual cortex, we know that there are columns of neurons that are oriented to lines of different orientations, right? So some respond mostly to vertical lines, some respond most strongly to horizontal lines, other ones in between. When you do fMRI, all of those are contained in like each voxel that you're imaging that's 3 by 3 by 3 millimeters. But just by chance, one of those voxels is likely to contain more neurons tuned to vertical lines, and another one's likely to contain more voxels tuned to horizontal lines, and so on. And the idea with the adaptation design is if you basically fatigue neurons that are tuned to one stimulus property by repeatedly presenting stimuli that those neurons are tuned to, over time they'll adapt, so they'll respond less strongly because they're seeing the same thing over and over again. And then if you present a line, so say you're adapting to vertical lines, then you present a horizontal line, other neurons are all of a sudden going to fire, and so that brain area should sort of show a steady decline in activation and then a boost again. Even though your individual voxels aren't necessarily capturing the individual tuning properties, overall you're sort of stimulating different populations of neurons within that brain area. So very simply, the adaptation design and the way Grille-Spector and colleagues originally showed it is just manipulating the number of different stimuli within a block. So the sort of strongest adaptation should occur when you just show the same stimulus over and over. And then as you alternate, here's just 2 different stimuli, 4 different stimuli, up to 32. There were 32 images in a block, so this is basically each image is different. And then look at the strength of the response. Sure enough, within lateral occipital cortex, which is an area that's responsive to object identity and object recognition, you can see that like the 32 stimulus block here shows the strongest response, whereas the 1 stimulus block shows a much weaker response, and the other ones are in between. And so when you sort of average that across subjects and across runs, make a nice bar graph, here you see the strength of the signal. So if we say the maximum signal's in the 32 item condition, that's 100%, whatever magnitude, it steadily decreases down to about 40% of that maximum as you decrease the number of different stimuli and increase the amount of adaptation. So you can use this in different kinds of designs to really tap into what properties of a stimulus is a particular brain area tuned to, or in a blob that responds sort of universally to a particular stimulus type. So say in language, you have these big blobs of activation. If you adapt to, say, by presenting words that all rhyme to each other, then you could sort of suppress the activity in areas that are sensitive to the phonology of words, but not to the semantics of the words, because the rhyming words all have different meanings. So you can use it in different sort of creative ways to get at that kind of thing. And then the last slide is two more modern sort of approaches, often used together actually. So the condition-rich design is basically saying, rather than just lumping all our stimuli in as like, these are all faces, and these are all cats, and these are all cars, or whatever, we actually code each individual stimulus as a unique sort of category or type. And then that gives you a lot more richness in terms of how you analyze the data later, because rather than just sort of saying faces versus whatever control stimuli, you could start looking at, well, what's sort of common or different between these faces. So you could pull out like male versus female faces, old versus young. You could look on an even more graded fashion at, different intensities, brightness, or whatever. So you can look at all kinds of different properties and characterize them. In language, we do this a lot because we're often interested in things like individual words. They might all be nouns. You might compare nouns to verbs, but all the words also differ in like the length of the word and how often they occur in usual sort of conversation or written forms. How imageable they are. They've all got these different properties that all vary on continuous scales. So we could just sort of say noun versus verb, or we could present a whole bunch of words and say, let's see how brain activation is modulated by lexical frequency versus familiarity versus imageability versus X, Y, and Z, right? So it gives you a lot more flexibility. It's in principle less sensitive, because basically now you're saying like, this one word is the one stimulus that represents a lexical frequency of 110. And this other word is the one stimulus that represents a lexical frequency of 240. So you assume that it's frequency that's sort of modulating things, but there's a risk that other things are playing in, or that you've got more random noise. So there are pluses and minuses. The time continuous design is another approach in which you just vary stimuli continuously over time. So the original studies doing this just had people in the scanner. They just watched like a 2-hour movie, and they were scanned continuously. And then they did actually look at things like when there were faces in the movie. It turned out face areas were activated. So you could sort of guess what they were seeing or what was in the scene by which brain areas were activated. But they also just looked at what was consistent across all of the subjects, and figured that what was consistent was brain activation that was driven by the movie, by the stimulus, rather than anything else. And then you can look at things like which parts of the movie are more interesting to people or more emotionally arousing or something like that. So this has become more and more common as people start using more complex stimuli and just seeing how things vary over time rather than trying to artificially sort of chunk things. So it gives you a lot of flexibility around much more naturalistic stimuli, watching events or different actions unfold and that kind of thing.